coordinator on NCBI's education team. Before coming to NCBI, I did computational chemistry research at Virginia Tech, where I focused on studying the dynamics and biophysical properties of non-canonical nucleic acids. And I did that with polarizable molecular dynamic simulations. Mostly my experience is with the Charm and Drude force field. So that's my background and my expertise. I'll be around, happy to talk about all that throughout the week. And if you'd like to connect, I'd be more than happy to do that. But for now, I'm gonna give a short presentation about how you explore 3D structures with iCn3D. And another disclaimer I wanted to give before getting started is I actually recently had oral surgery. So if at any time my speaking is a bit muddled or unclear, please just let me know and I can try to, slow down, speak up, whatever. I want to make sure this is a good experience for you, and that might get lost in the time. All right, so the first thing I want to do is align expectations a little with this presentation. So this presentation is fairly short, and as a result, we're only going to have a time for a couple things. What we're going to do is a quick introduction. I'll demo iCn3D and help you get oriented in the tool. That'll include selection, coloring, and styling. We'll look at some interaction networks, mutation analysis examples, and then I'm going to point you to some materials for continued learning before I pass it over to Henry for the next presentation. So we have a lot of people here, including Xiao, who's a developer for iCn3D. People can jump in and answer questions at any point in time. Be great to keep some of that in the chat just so we stay on track. But there's a lot of expertise here, so don't be shy to fire away some questions. I'm sure we can get it answered. And then keep in mind that this presentation was originally generated for a crowd with limited structural biology and visualization experience. What we're trying to do is introduce you to the power of iCn3D, get some of our participants up to speed, start having conversations. So if you feel like you're an advanced user in iCn3D or molecular visualization, certainly you can stick around and help answer questions in the chat, or you can get up, grab a snack, and just sit tight for 30 minutes before the next presentation. Just want to be upfront about that. And then the last thing I wanted to say is we don't have time to cover all things structure related, obviously. This presentation was one that I adapted from a longer workshop that was 3 or 4 hours. So I had a lot of supplemental materials that I just kind of shifted to the back for you. So I just want to let you know that that's there. This slide deck is in the presentation Slack channel. So definitely flip through that, take a look at those supplemental materials. It'll range from some fundamental information on concepts of DNA structure, nucleic acid structure, protein structure, and then go into some more in-depth information on, structure determination, experimental and computational techniques. And I'd be happy to talk about that a little bit more offline. But for now, I think we should just jump right into this. So for those of you who don't know, iCn3D is an NCBI tool that was developed and is maintained largely by Dr. Xiao Wang, who is an organizer here who you've met. And Codeathons like the one we're kicking off now have been really instrumental in tool development and improvement. So just know that all your work and your feedback this week and today really does go a long way. iCn3D is a free web-based tool which is supported by NCBI that allow you to explore the function, the structure of a biomolecule at an atomistic level. And like some of the people we've talked to today, there's a lot of functionality in there and it connects you to information in ways that some other structure software does not. I think the most easy way to explain iCn3D to most structural users is it's somewhat comparable to PyMol, VMD, and Chimera tools, but it's connected with a lot more information at the NCBI structure database. It's web-based, which means no installation is needed to use it. That can be really great if you're teaching or if you're doing an event like this. You don't need to download structure files, which is actually really great if you just are kind of trying to pop through some information fast. You don't have to do a bunch of downloads or get a messy download folder or anything like that. And then there are a ton of other cool features that you'll work with this year and we can, or this week, and we can explore a little bit. But just broadly speaking, in iCn3D, users can visualize 1D, 2D, and 3D structure. You can view information about the sequence and structure alignments in the tool, all while working with the 3D structure, which is great. You can probe mutations, Another great thing is you can really easily save and share links of your customizable display. That's really easy to do in iCn3D. If you're collaborating with people or if you're teaching novice users, this can be a great way to work together. And then iCn3D has so many other features that I'm sure a lot of you are aware with. Some of these are easy use in Jupyter notebooks, 3D printing capability, great contact maps, pre-calculated symmetry, symmetry dynamicality, electron density maps, protein analysis, solving accessible surface area, and more. And development is ongoing, so this list will just keep getting longer, which is great. And then in fact, Xiao will be presenting soon about new features of the tool, which include virtual reality of 3D structures and batch analysis. So definitely talk to him about that, connect with them, potentially go to the presentation if you're available. I'm sure he'd be more than happy to talk about those things. So as I mentioned before, iCn3D is connected to the structured database, which is derived from protein data bank records, but also has additional 3D information, including chemical graph info, validation of secondary structure, and taxonomy. It also connects 3D data to all sorts of information housed at the NCBI, like literature, molecular data, chemical data, and other tools. And in my opinion, it does this better than a lot of other commercial softwares that are out there. So now I think it's good to just jump in, get started with iCn3D. This is going to be something that's is more of a hands-on activity. I know that there are people who are, sitting in a room and working on this. It might be hard for you to ask questions. So if at any point in time, you're confused, you need things to slow down, please let someone maybe let Ravi and Tom know, and we can try to address that. One thing that I wanted to show before we jump in is this slide with iCn3 shortcuts. This is really helpful while you're exploring the tool. This was just pulled directly from the help document, which is linked in the bottom right. I just wanted to introduce that to you, before we jump in. You can use this slide or, of course, the help document for resources during our exercise or during the Codeathon week. So let's get started using iCn3D. I tried to make these slides easy for the audience to follow along with. So just to give you some background, these highlighted sections are going to be actions that you take, things you click on. If you're following me, just keep that in mind. And then at the bottom right, you're going to see a link. If you get lost or fall behind, clicking on a link will take you to the structure rendered by the steps in the demo. So pretty much just clicking on that will take you to the step I'm at the end of the slide. And those will be updated as we flip through the slide. So you can get to iCn3D with a direct link like this. You can also Google it or get there from NCBI structure database. I'll show you all those ways. And then just one moment because the Zoom screen is like blocking me from pulling things up. All right. Is everyone able to see my web browser here?
Yep.
Cool. So this is what happens when you go to that link and you want to hit biological unit. Another thing that you can do is go to the structure database. For those of you who do primarily searching in PDB or something like that, this is somewhat similar. You can search several terms to get different structures. But today I'm just going to go to one of the most standard examples, which is PDB code 1TUP. When you put in a code that is specific, like a MMDID or a PDB ID, it's going to take you right to a structure summary page like this, which has all sorts of really great information. Really encourage you to check this out, especially, if you're working on one biological concept, flip through here and try to understand all the information that's there. That's a great starting point for any project. If you scroll down to this molecular graphic, you'll see full featured 3D viewer. Clicking on that will pop it open for you in iCn3D, which is great. And then, as I mentioned, you can Google this and get here pretty easy as well. So I'm going to close sequence and annotations for now. We'll come back to it. So the structure I'm using as today's example is PDB code 1TUP, which is a tumor suppressor P53 complex with DNA. When you pop it open in iCn3D, you can see the biomolecule has 3 protein chain. It has duplex bDNA there in the middle, and then it also has different zinc ions. Those are all colored differently in the tool, and it's rendered in this chain style automatically, but you can go through and change that. Before we start poking around in iCn3D too much, I do want to point out there is this help button, which is, as it suggests, very, very helpful, especially if it's new. If you're new to this, I would suggest trying to flip through selection hints. Maybe if you have a little bit more time, doing some tutorials, but there's so much information within this help drop-down menu that I really, really encourage you to check out. So if you have this open like I do right now, and you've never been in iCn3D before, I just want you to play around with it. It's pretty intuitive, but you know, if you're familiar with maybe Pymol or something like that, and you have that muscle memory, it might take a little bit of time for you to get used to it. But of course, you can spin around, you can zoom in really easily. If you hover over the structure, it'll tell you right now what residue you're looking at. And I don't know if you're on a Mac or a PC, but if you want to make a selection on a PC, hitting Alt and clicking on it will do that for you really easily. So you can select what you want, and then just make sure you pop that selection button off if you no longer want to use that selection. I think that's a common mistake for people who are new to iCn3D, is to, you know, accidentally select some stuff and forget what they're working with. So just keep in mind that button's there and turn it on and off as you need. So as I mentioned, when you hover over the structure, some information is returned, namely just what you're looking at, but you can change the type of information that's returned by going to select, and then select on 3D. And then a popular one to switch to is to change it to atom view. That'll just give you a little bit more specific information about what you're hovering over, and it'll tell you what atom you're over specifically. So the rendering that we have right now, the style is cartoon. So by just looking at it, there's not a lot of atomistic details you can discern from it. So a way to change that is to change the way that this is styled. And the way that you want to do that is go to style. And first, let's change the side chain to the protein. I think that's the most intuitive step. So if you go to side chains, you can switch it to lines, stick, ball and stick, sphere, whatever you fancy. I like lines because it's not super messy. And then now you can see the side chains of your protein. And when you hover over it, you can see exactly what you're looking at. And that can be a little bit more helpful. So I think that the style buttons are pretty intuitive, but I just want to pause for a minute or two, allow you to flip through the styles that are there, render this in a way that makes sense to you, and then we'll move on to coloring really quick before we do some more selection and mutation analysis. What I like to do, this is pretty close to how, when I'm new with the structure, this is pretty close to how I would like to look at it. I like to keep my protein and ribbon, side chains as stick, and then nucleotides and cartoons. If you're just trying to get an overall view of the structure is good. You might want to switch nucleotides to something more like ball and stick if you want a closer look at what atoms in the bases or backbone you're looking at. So again, just going to pause for a minute. Please send messages in the chat if you have any questions, or just speak up, and then we'll move on to coloring. And Ravi, since I'm not there in person, could you just let me know how everyone's doing in the room? Is this coming across okay and people are able to follow along?
Okay, let me get the feedback.
Okay.
Any questions here? Okay. I get all thumbs up.
Cool. Cool. We're at the beginning, so that makes sense. Another thing that I want to point out is that at the bottom, this is tracing the steps that you're taking. It's writing it out. So that can be helpful. You know, sometimes if you accidentally select the wrong thing, you can see exactly what you did down here in that little bar, and that's helpful. So if we don't have any questions, I do want to just plow right through and get to coloring so that we can get to more advanced stuff. So like style, color is an important selection for your understanding of structure, and it's important when you're communicating, you know, aspects of your science to the public. So as I mentioned before, the default coloring is chain. As the name suggests, it's just going to color each chain differently. The coloring in chain is what you see in that 2D map on the structure website. So, you know, that can be a helpful thing to check out. I believe Philippe showed some of that in his slides too. So just keep in mind those are consistent. Another popular option for coloring, especially for people who are kind of newer to molecular visualization, is rainbow. So what rainbow does is it colors the N term or the 5' N as red, and that flows to blue for the C term and the 3' end. So when you're looking at a structure with rainbow colors, you know that the warm colors are nearer to the N terminus or the 5' end, and then the cooler colors are nearer to the C terminus and 3' end. I don't personally think that this is a great coloring selection for a structure like this, but maybe if you have a globular protein or something that's very symmetrical, and you're kind of, you know, losing orientation a little bit, that could be a good way to do that. And then of course it's a little bit more helpful if maybe you select different components and not a whole big structure as this color. But it's good to know how that works, because like I said, a lot of people, especially when they're new to molecular visualization, like to use rainbow. So it's good to know what's there. Another coloring that I like is charge. I think that not only is this visually appealing, it gives you some quick information. It colors positively charged as blue and negatively charged as red, and then neutral aspects like carbon as gray. Similar to charge, if you go to atom color, that's another one that I think provides similar information and is pretty visually appealing. What that does is carbon is this like gray or white color. Oxygen will be red, nitrogen will be blue. Those little yellow pieces are sulfur. So that's another good option for you. And then there are all sorts of different color, more advanced coloring options that I think are pretty helpful. Briefly, secondary coloring can be really good to get some quick information. Another one that you have is hydrophobicity. That can provide some really good information. And then solvent accessibility is another thing that you can look at really quickly. And you can change the percentage cutoff here and then get some quick information about your structure, which is great. Once again, I just want to pause for a second and encourage you all to flip through the colors, find one that you feel comfortable with, and then we'll move on to selections. So I think I'm going to give us 2 minutes to flip through colors. Maybe pick a style that you like.
You can go to define sets.
I think I'm going to stick with atom. That's one that I tend to like when starting.
And then you can.
And I see we got some questions in the chat, so let me look through those.
I think, Alexa, this might be a matter of what we're used to. So for 20 years, we used CN3D that had rainbow coloring 5' to 3' or amino to carboxyl, Roy G. Biv, you know, red, orange, yellow to violet, right? And it just got added back to iCn3D, and it's not behaving the way we used it for 20 years. And so we're having some debates about how it should be behaving.
In fact, go to the menu and apply rainbow by chain. There you go.
Yeah, that's how it should be.
That now conveys a proper biological representation. And so one of the things, spectrum and rainbow were modeled after pi mole. this is one of the problems with other programs, is they treat a structure from head to toe, and they just put the sequence in a line, and they go 1, 2, 5,000. And actually, biology assembles chains of molecules together. And so these are actually a meaningful representation.
And the reason we use rainbow with DNA is to convey the anti-parallel nature.
Yes.
Because students get really confused about how can it be different directions, or, so that's why we use it.
And
Yeah.
proteins actually are fascinating when you get into like the phage proteins in a phage coat. They'll assemble in a parallel fashion. And so you get beautiful, in rainbow, you get just beautiful spiral tie-dye kinds of representations that you can put on t-shirts and stuff. It's awesome. It's
That's great. That's a great point. Looking at parallel versus anti-parallel or mixed structures is pretty good for rainbow coloring. I did work with G quadruplexes. So yeah, that kind of stuff was really important to understand how the core worked.
Jai, I was smiling because we've had this debate.
Yeah, Gio, by the way, we should actually lump together spectrum and rainbow and just have a toggle so that, people can go from magenta to red or red to magenta the way they want. Because right now, we have discussions we should have. But I would answer also that we didn't model after PIMO, okay? We modeled after the rainbow. The 7
No,
colors,
but so
okay?
see, yeah. Sorry, I was cutting you off.
And then it's a matter of following an order. Now, what I would point to something very, very, very interesting and important with this kind of coloring. Yes, you should do it chain by chain or domain by domain, because then you learn how this folding goes on. It's very useful. But it can be a long debate about color. So let's stop here.
Oh, I'm neutral on spectrum rainbow. I mean, having both is great. It's what you just said. It's chain by chain, domain by domain, because that conveys structural information and folding just using color to visualize it.
By the way, here to the people who don't know much about proteins, this was very interesting when I first started molecular graphics. We had only 300 proteins in the database. And so I could look at all of them, as I was fortunate to have my Evans and Sutherland at the time, and then Silicon Graphics. Now, precisely, the very interesting thing about domains, and when you use color, you can see that easily, is that your domains fold with either the N and C terminus far away, or with N and C terminus coming together. Mm-hmm. So with color, you will spot immediately the kind of domain you might have. And if these domains will actually get piped into each other sequentially, if you want, forming very long chain, or if they actually will be more like plugins for each other when the N and C terminus come together. So the coloring is extremely interesting, and definitely when I say we're going to have to go towards automation, we're going to have to make decisions on the kind of coloring we will pick up, the kind of style we will pick up automatically. Because right now, it still requires that you make a choice. But for people who don't know all these things, it will be tremendously useful to understand, by color and by the choices we make, what the protein looks like. Well, what one representation looks like that might become a general one. Sorry, too long.
Yeah, no worries. And I mean, these conversations are exactly why we have events like these. If you want to continue them in the chat, that's perfectly fine by me. I just want to move forward a little bit so we can get through some more stuff and I don't run over time. So another obviously really important aspect of iCn3D or any molecular visualization software is the ability to make selections and make changes to part of the structure, which is what some people have been chiming in the discussion is. So if you're new to iCn3D, I think one of the easiest ways to get started with this, besides just holding Alt and clicking on things, which easier said than done to click on the right thing sometime. So one way to do this is to go to Analysis and then Define Sets. And what that does is it's showing you sets that are already defined in the structure, such as this is chain A of the protein, B, C. You have ions, you have nucleotides, you have all the proteins together, water, if it's applicable to that structure. That can all be just a really quick and dirty way to change aspects of the structure. So for this, for example, I don't really like the duplex DNA being all red. So I can go ahead to color and I can go back to rainbow, which we've been talking about, and do for selection. And then that's changed. Again, just pay attention when you're done with the selection you're working with. Make sure you pop that off and make another selection. It's really easy to, forget about that. So you can select those components based on their chain names or properties. Another good way to make selections pretty quickly is that sequence and annotation buttons that I closed out at the very beginning. So if you go to Analysis and then you go right down to Sequence and Annotations, there's a whole host of information here, which is one of the powerful ways that iCn3D connects to other information that things like maybe VMD, Polymol, and Chimera don't always. So I definitely, this week, explore everything that's in the Sequence and Annotations buttons. While you have all these experts here, definitely ask questions. But if you're brand new to this, one thing that I might suggest is just clicking off everything and going to details so that you can just see sequence information and highlight things there. I just want to show you how that works. When you highlight it, then you can style it. Maybe you want to show that stretch of the sequence a little bit more and stick, so you can go ahead and highlight that. This is just kind of nonsensical right now. I'm just showing you how that works. So I'm going to turn it off. And then we're going to talk about selecting by distance, which is one of the things that I feel like when I was doing my PhD, and especially, if you're working with simulation data, selecting by distance was like one of the most common things I did. So I think that that's a pretty important thing to talk about. So for selecting by distance, I'm going to keep selected sets open. Honestly, just the defined sets, I like them. I think it's really easy if I misclick to have this here. For select by distance, right now for this example, let's take a look at the residues that surround these zinc ions so that we can get a better understanding of the ion-protein interactions there. So to do that, I clicked ions and selected sets, but you can click that over here. You go to select by distance, and then it tells you to select your first set and your second set. For one, I'm going to keep selected. I think you can also hit ions here instead of having it selected there. But when I was practicing this a little bit this morning, that was being a little bit finicky. So I'm going to stick with this for now. Let's give it a little more permissive of a radius, 5 angstroms, just kind of arbitrarily, to make sure that we're looking at all those residues around. And then you can hit display. And you can see it just kind of highlights everything that's around those ions. This isn't the best way to look at it right now, so let's do some stuff to improve that. One thing that I love to do, especially if this is something you're going to be working with multiple times, is just save that selection right away and give it a meaningful name so you can come back to it. So what I'm going to name this selection is Resi 5A from zinc. So to me, that just means it's all the residues 5 angstroms from the zinc ions. That makes sense in my brain. That's what I'm going to name it. And then that will come over here to the define set. So if I accidentally close this, I misclick, whatever, I do something, I can always just come back to the save selection here and not have to repeat all the steps, which is nice. So if we want to have a better view of this, I'm going to click the selection that I just saved. I'm going to hit Control and click ions as well. And then I'm going to do view and view selection. And then you can get a better look at what's going on there. One thing I did forget is I probably want to style the 5 angstroms from the zinc a little bit better so that they stick out. So I'm going to click on my define sets once again. I'm going to go to style, side chains, and just stick so that you can see them a little bit better. They're a little more obvious. And then let's do that again where we look at those together. So yeah, that's just a quick and dirty way to look at any sort of specific interactions. Yeah, it can be really helpful if you're new to working with this type of software to know how to do that. So I wanted to show you that. So what I want to do now is I want to show you that, first of all, you can click out of that really easily, and then you come back to your full structure. You could also hit view and view full structure there. It would do essentially the same thing. But I want to give everyone about 5 minutes to just play around with selections, colors, style, build on the 3 things that we've kind of talked about, render this structure or a structure of your interest in a way that you think is meaningful. And then if you like what you have, like I said, we have a lot of experts here. So if you want to share that interactive link in the chat, we can all take a look at it and learn from each other. So just really quick, the way that you want to save things is you go File, Save Files. You could save it as a PNG image if you'd like, but right now, I think the best way to do this would be to share link, and then it'll create a link for you to share. Didn't seem to.
It looked like it was saying you had more than 4,000 characters in your link.
Okay, so I'd probably just have to refresh and not do so many things then.
Yeah, yeah, Shayao, isn't it saving like the whole session?
Then you can save the PNG image for now.
Yeah, so it works. Yeah, as they mentioned, so the way that the shared link does is it tracks all the steps that you've taken. So for me, while I was flipping around with all the different styles, I'm taking a lot of steps. So it probably, if I like really loved what I was showing here and I wanted to share it with my PI, someone I was working with or something like that, probably just be easier to open it fresh and take a more direct approach to getting the structure, and then you can share the link really easily. I'm sure there are other ways to do that too, but for
you can share, still share, but you would have to send the script. I mean, if you go to, I mean, GIO can tell you that if you go to File, you can save it as a PNG file, and then you send the file, and the guy can open it. Now, ideally, if you work on the same website, you can put it on your web server, and then you can click on that file on the web server. So that's something that definitely we need to improve, so that we can make longer analysis shareable. Right now, 4,000 characters is the limit.
And then this is only an issue right now because I think Zoom doesn't support sharing pictures. So I was just going to say share a link. But yeah, you can save it as a PNG image really easily. One thing that you might want to do for those of you who are new, if you're saving this for, I don't know, to cut out and put in your lab notebook or to put in a report or something like that, make sure you change the background from black. And you can do that from style, background, and then probably just want to change it to white. I think black works when you're working with it on a screen, but certainly if you want to render this for, you know, a report, a publication, something like that, you want to turn that off. So let's just do 2 minutes of playing around, and then I'm running short on time, so I want to show a mutation analysis, at least one of the examples really quick, and then point you to educational materials.
Alexa, while we're doing that,
Yep.
the saved selection, is that now appear in the defined sets?
Yes, it will.
Okay.
This is the, I'm sorry,
Okay.
my screen's kind of big, so it might look small to y'all,
Yeah,
but
yeah.
that's the Resi 5A zinc. So
Okay.
yeah.
Good. Thank you. Yep.
Thank you for the question.
Yeah, I didn't know if you had shown it or, I mean, I may have not been paying attention for a minute.
Yeah, sorry, I'm flying a little bit because
Oh, no, don't worry about it. Yeah.
we're trying to show a couple things and we're having good conversations, but I don't want to take away from the important time that the teams need to meet and stuff. So, okay, I want to really quickly just show a mutation example. I actually have 3 different examples in the slide deck with answers in the slide deck. I'm not going to get to all 3 of those, but definitely if you have time to try this out on your own, do so, ask me. I think it sounds like Ravi's working on some ways to improve how mutation analysis is done in the iCn3D. But I do think that this is a really cool and quick way to look at how small structural changes might influence things like protein DNA interactions. This is pretty similar to PIMO mutagenesis for those of you who are like native to PIMO or something like that. Very powerful tool. I think iCn3D just makes it really easy. So this is one of the favorite things I show people who are like undergrads getting started with this, just so they can understand, you know, practical application of some of the analysis in iCn3D. So before we go to mutation analysis, I wanted to briefly talk about P53 binding and provide a little bit of context about protein DNA interactions. So P53 binds to many regulatory sites in the genome, and what it does is produces proteins to either halt cell division until damage is repaired or initiate apoptosis if damage is too severe. The protein is rich in positively charged amino acids like arginine, histidine, and lysine. And in terms of protein DNA interactions, positively charged amino acids commonly interact with that negatively charged backbone of nucleic acids, as well as within the major groove of the helix or in areas that are more solvent exposed. So those are just some things to keep in mind when we do this exercise. So let's pop back to iCn3D to check this out. Since I did have the issue where I was clicking around with a bunch of different things, I think I'm going to start with a more fresh link just to make it a little simpler. So this is just a simple view of the structure. And if you have the slide and if you want to pop that open, you can just click on the link in the slide. So, okay, using what we know about structure and protein DNA interactions, let's try to find residues of interest with the molecular visualization tool. So how might we do this? One thing that we can do is select by distance and see what's within hydrogen bond cutoff from the DNA. So to do this again, I would keep your defined sets open, but let's go to select and select by distance. Again, it was being a little bit finicky earlier, so we'll see if everything works. But the first selection I want to make is going to be nucleotides. And then I'm going to drop this cutoff radius to 3.5, which is roughly the hydrogen bond cutoff. And then let's just focus on protein chain B for right now, so that we can look at the interactions that are there. If we hit display, it looks like it should highlight some residues that are interacting. And again, I think it's good practice to save this selection. I'm going to use a similar naming convention, which is Resi 3.5A to DNA. Hit save, so it comes over to the selected set again. I am going to style this in stick, so it's a little bit more obvious what's going on. And there you can see how those side chains are kind of reaching into the DNA and helping, you know, maintain the DNA protein interaction. If we want to take a closer look at how this looks, you can again select your selection, hit control, select nucleotides, and then view selection. And you can look at pretty much exactly what we just defined. And if you kind of hover over this, you're going to see those are some arginines, a cytosine that are interacting, and then lysine 120 that looks like it's sticking in pretty far. So that lysine 120 residue does pop out to me as interesting because it's pointing into the major groove, and we know lysine is a positively charged residue. So my mind thinks, okay, what would happen if this residue was disrupted? And we can actually test that pretty easily with iCn3D by going to Analysis, going down to Mutation, and then this pop-up shows you exactly how to specify a mutation in iCn3D, which is really handy. So for this, we're going to go our PDB code 12UP. This is the chain ID, which, like I said, we're working with protein B right now. So I'm going to do B, residue 120. And then I'm going to switch that lysine to an alanine because that changes the charge interactions. And actually, if you look at the structure paper that's deposited with this, one of the biggest things that they discussed is that a lysine to alanine mutation at point 120 leads to a disrupted DNA protein binding in their mouse models. So this is something that is biologically interesting. So when you specify that, again, 1TUP, B, 120, and then mutating it to A, you can hit interactions. It's going to open a new tab for you. It'll tell you, press A to alternate between wild type and mutant. You hit OK, that's just good information. And then you can flip through, look really closely at the interaction in the wild type, and then hit A to see how that interaction would change if the point mutation we specified happened. So feel free to toggle back and forth and kind of get a visual understanding of how that might work. Obviously, it looks like it's a smaller side chain, so it's not reaching as far into the major groove, but it looks like there's a lot of really important contacts that are disrupted, which of course could disrupt binding. But if you look over here at the interaction panel, it'll actually tell you exactly what changes. So this key up here shows green as hydrogen bond, cyan, salt bridge and ionic, gray as contacts, magenta halogen bonds, red pi cation, blue pi stacking. So that's all really great information. It has some 2D maps to see what that looks like. And if you scroll all the way down to the bottom, there's this helpful one that's different interactions, and it will pretty cleanly show you what's different when this mutation exists. So you can get some quick and dirty information about how altering this residue might alter the important interaction. And I think that's really helpful and powerful information. A disclaimer to think about, as Ravi's project is kind of touching on, is that sometimes when you have a point mutation, that influences folding. And so the interaction might not look exactly like this. But in terms of the type of information that you can provide pretty quickly, I think this does a good job of following like basic scientific and biological concepts to understand how things are working. And it kind of highlights the power of working with structure and tools like this, and emphasizes the importance of why we're having meetings like this to improve said tools. So I'm 3 minutes over on time, so I'm just going to fly to the end and give a couple last statements, and then I will turn it over. So as I mentioned in the intro, we've really only scratched the surface of what iCn3D can do, and I encourage you to keep playing around with the tool, using the tool, improving the tool. I feel like I'm like talking to the choir a little bit, but you know, this is all really important stuff. And there's a lot more resources for learning that people like you and Zhao and Philippe have put together. So I really encourage you to check those out and point other people to those resources as well, because there's a lot of great information there. And then lastly, I just wanted to give a plug to NCBI education. There's tons of resources at the NCBI, and we offer free virtual workshops pretty regularly. You can check those out at our NCBI Insights blog and apply to them. We're working on a more advanced iCn3D offering probably in the beginning of 2023, but we talk about all sorts of things like basic coding in Python, virology, stuff like that. So for people like you who are passionate about open science, just definitely keep an eye on that page because there's a lot of stuff going on in the education team that I think might interest you. And with that, I want to stop talking and turn it over to Henry, and I'll answer any questions I can in the chat.